Hello, everyone, and thank you so much for joining us today, wherever you are in the world. My name is Sarah Jane Demy, and I am the CEO of Demy-Colton, located here in New York City. And it is my pleasure to welcome you to the 6th installment of the Demy-Colton Virtual Salon Series. As many of you know, Demy-Colton's mission is all about convening biopharma and digital medicine industry leaders to catalyze change. We know we can help drive innovation when we bring individuals together who are committed to and united by a common cause and a shared passion. And as you know, in our industry, that passion is bringing cures and therapeutics to people in need. We were in the process of launching our new business development and investor conference in New York called BioFuture when COVID-19 changed everything for us, as it did for many people around the world. So in lieu of an in-person BioFuture meeting, we launched the Demy-Colton Virtual Salon Series to bring great content to you and continue our mission of bringing people together with a shared passion. I would like to take a moment to recognize and thank all of our sponsors who have stood by us during this challenging time, and indeed who have also broadened their support as our programming has evolved. At Demy-Colton, we believe the Virtual Salon Series will be here to stay, even after we get beyond COVID, which we hope is very soon, and go back to a more normal life, if you will, whatever that new normal will be. Please do check our website for the schedule of salons, fireside chats, topics, and participants. And of course, I encourage you to join us at future salons. Before introducing today's panel, I just want to take a moment to briefly touch on the logistics of our session. We will go until about 10 to 12 with the discussion. And after that, we'll end the formal portion and move on to the Q&A. We'll do that for about 15 minutes. And if you'd like to ask a question, on your lower portion of your screen, you'll see a pane that says ask a question. Please submit your question, as we're going through the discussion, and we'll get to as many as we possibly can during the Q&A session. Please also note that this salon will be recorded and available for on-demand viewing later today. Additional materials can be viewed in the Event Resources tab as well. Now, today's installment of the Demy-Colton Virtual Salon Series is Futurescaping of Oncology, a technology sampler of potential paradigm-changing approaches to treating cancer. I'd like to introduce our panelists. First up is Dr. Jeffrey Bachman. Jeff is Executive Vice President of the Oncology Practice at Cello Health Bioconsulting. Previously, Jeff was a Senior Research Scientist developing oligonucleotide therapeutics for viral diseases and cancer at Innovir Laboratories. Prior to that, Jeff was an Assistant Research Professor at the George Washington University School of Medicine. And Cello Health Bioconsulting is a knowledge-based consultancy deeply rooted in science and ever-evolving challenges and opportunities facing the biopharma industry. Jeff will serve as our moderator today. Our next panelist is Dr. Jake Becraft. Jake is a synthetic biologist and the co-founder and CEO of Strand Therapeutics. Together with colleagues at MIT's renowned Synthetic Biology Center, he led the development of the world's first synthetic biology programming language for mRNA. Next up is Dr. Jennifer Petter. Jennifer is the founder and CSO of Arrakis Therapeutics. Previously, she held senior positions at Celgene, Avila Therapeutics, Mersana Therapeutics, Biogen, and Sandoz Novartis. During her career, Dr. Petter has ushered multiple compounds into the clinic for the treatment of cancer, cardiovascular disease, autoimmune disorders, and sepsis. And last but not least is Dr. Arthur T. Sands. Arthur has served as the CEO of Nurix Therapeutics since September 2014. Prior to joining Nurex, Dr. Sands was the co-founder, president, and CEO of Lexicon Pharmaceuticals, a company focused on target validation and pharmaceutical development from 1995 until 2014. Before founding Lexicon Pharmaceuticals, Dr. Sands was I forget what you were. I didn't write that down, Arthur, so please forgive me. I'm sorry.
It's all right.
Forgive me. With that,
postdoctoral fellow.
Sorry?
I was just a postdoctoral fellow before I got into biotech.
That's right.
I was nothing.
Well, you were becoming something.
Wonderful lead there.
Yeah, I know. Great segue, right? We hope all of you will join us when we do our in-person BioFuture meeting, which is currently scheduled to take place in April of 2021. We hope that will happen. And without further ado, Jeff, I'd like to turn things over to you to lead this great discussion.
All right. Well, thank you, Sarah. Thank you, Demy-Colton. Thank you, everyone, for attending today. I hope everyone is doing their best to stay safe amidst COVID, amidst fires, amidst whatever else might be going on in this very strange 2020. As you can see, we're all in our respective homes or offices. Jake, that's a nice plinth, or a version of a plinth behind you. So, all right. Not the original. All righty. So we, as the title suggests, what we're trying to do is use these three different technologies and these three different biotechs as exemplars of the opportunities and challenges, and hopefully some of the potential, paradigm changes that could ultimately impact cancer management and improve the lives of patients. So with that, again, although we will focus on each of the companies and their respective technologies, we will try to broaden our thoughts and comments to kind of the greater implications that some of these issues have for many of you in the audience who are either nascent biotech or investors or perhaps biopharma entities that are looking at licensing or innovative technologies. So just to start us off, as one can see, there's a little bit of a theme here between Rox's and Jennifer's company with kind of a small molecule, so validated modality, but targeting RNA. We have Jake with Strand with the idea of using RNA as a therapeutic, and that implies the potential for kind of multi-component therapeutics, and we'll touch on that. And of course, Arthur with Nurex and a host of many other companies are trying to exploit targeted protein degradation and go after a variety of more problematic, and that's probably true for everybody. But each of these companies are in slightly different phases of their life cycle and building out their pipeline. So what I want to do is pose the question that is germane to anybody at these stage of early stage pre-commercial, pre-POC companies, whether that's germane to their own business strategy, germane to prospective partners, germane to investors, or in the case of public companies like Nurex, investors, the market. How do you go about thinking as how you start to build out your pipeline, even very early for a company like you, Jennifer? You have to be starting to tell a story, and so thinking about what is unique about your platforms, and how does that enable you to think about what you want to target. So maybe we'll kind of go in sort of order of stage of maturity, if you will. So Jennifer, we'll start with you. Not the person, for company.
I understand. We do concern ourselves with our discovery portfolio. We do look further down the road to try to understand what the potential clinical problem is that we're solving, and therefore, by implication, the commercial implication. Yes, you have to look far enough down the road to make sure that the problem you're solving will not be solved presently by yourself. Having said that, the targets we're going after, which are these complex tertiary folds of RNA, are sufficiently novel. We have to focus on the underlying mechanistic validation as well as biological validation. So our approach in general has been to, in essence, minimize biology and clinical risk by going after messenger RNAs that express proteins we know be valuable, either because we know that in a clinical study or we know that pharmacology biology studies. In that latter category, we have to be mixed, where I think all of us believe that you could press.
Hey, Jennifer, it's Sarah. I'm sorry to interrupt, but we are having some trouble hearing you. You're breaking up.
Yeah,
and
maybe you need to get closer to Jennifer, like I did.
Yeah, lend that air of intensity.
Oh, so that's much better.
Okay. And so, I think that and MYC is an example of a biologically validated target where if you could suppress its expression with a well-distributed molecule, there's probably a drug in there someplace. So again, I think that we're very concerned about putting this together, but we do have to make sure that we don't pursue something that would be mechanistically unsuitable, which is still a very much undiscovered country for us. In trying to pursue something that might be commercial.
Great, got it. So Jake, how about from your perspective?
Yeah, so interestingly enough, I have a very similar outlook in terms of risk management as Jennifer was just saying. And actually, Jennifer and I have had conversations about this before, and I've probed her deep expertise about risk management in terms of target selection. And so where we're coming from is a different sort of viewpoint, though, of technology, where small molecules are, as you said, Jeff, more de-risked, at least as a platform or as a type of drug, whereas we're dealing with a fairly novel modality to deliver, which is the synthetic messenger RNA. And so when I think about how to manage that risk, if we're starting with a novel modality, then the genes that we deliver and the mechanisms that we target with those proteins that we're delivering encoded on the messenger RNAs should be things that are already de-risked in the clinic, things we've seen. For example, cytokine therapies have had all sorts of high levels of efficacy in treating solid tumors. But one of the huge drawbacks of those cytokine therapies has been, the pharmacodynamics and the PK and the degradation of the protein therapeutic and its systemic leakage, outside of the tumors and systemic toxicities. And so can we perhaps take a mechanism that we think is fairly de-risked, something like a cytokine, and repackage that into or redeliver it in a new way that is secreted directly by the tumors themselves, so that you have this much higher local concentration and hopefully get less systemic toxicity as a response. But when I think about actually developing the pipeline and how do you manage that sort of risk with a new modality like mRNA, my goal has been, let's get into the clinic as fast as we can. Let's get the first drug that we had been working on even at MIT before we started the company and now working to commercialize. Let's get that into the clinic to start to show what we believe is going to be a high level of safety and efficacy data, and that will help us build out a platform that will have, this kind of infinite platform of, any drug, any protein that we want to deliver, whether that's, something endogenous, whether it's an engineered protein that we work with pharma partners, whether it's antibodies or bispecifics or such that other people have developed and we can encode on messenger RNA. But in order to get to that point, in order to get to the point where we can tee up, 40 shots on goal with a giant pipeline, we have to get that first de-risking step. And that's why in the first 18 months of the company, we went from conception to first money in the door to beginning IND enabling studies. And we were doing manufacturing on day 60 of the company, basically working on getting everything up and running so that that platform can be as solid as possible. And then that allows us, as we raise our Series A and our Series B, to just tee up a whole plethora, a wave kind of drugs that we can build off of that first proof of concept. But it's a different approach because the platform itself is pretty much entirely novel. Though 2020 may be the year that we all get injected with messenger RNA.
Exactly. We'll see. We have AstraZeneca on hold right now. But
not a prediction I would have made at the beginning of the year, but we're seeing that the idea itself, I think, is very robust as far as its ability to deliver certain proteins, and it just opens up a place where we could partner and build with kind of everyone.
So, quick question for you before we go to Arthur. I'm going to kind of put you on the spot a little bit, so you might have to take the fifth, and that is your website, your company, and since I've known you even when you were just transitioning out of MIT, so it's pretty stealthy. So, whereas Jennifer, for example, referred to going after at least a biologically relevant protein mix, but going after the RNA, you've only referred generically to cytokines. Can you speak to whether you're de-risking by going after IL-2 or semi-de-risking by going after something like IL-12, or do you have to leave us waiting for more?
I have to leave you guys waiting for more for now on what exactly we're using. But I think the overarching theme is that ideally we'll be moving into very soon be moving into a whole number of different sorts of things that we can encode. And I think what's special about what we do at Strand is that it's not just about delivering the protein that is encoded. That is a piece of the mechanism. But as anyone who's heard me speak at synthetic biology conferences and about the engineering that we're doing, for us it's about a drug that's able to hit multiple different mechanisms, stimulate innate immune responses, stimulate an adaptive immune response, cause tumors to lyse themselves and present their antigens. And you can do that with a single protein, but you also need to program the RNA so that everything you're doing has to happen in concert, right? You can't immediately kill the cells when you go into them, or you'll get no protein produced at all. If you're trying to go into the tumor. And so it's this idea of programmability and control that we really harp on and is really key to the success of our drug and what I think will make it incredibly effective. Our scientific co-founder did publish a paper in Nature Cancer about a month ago, and everyone can go and read that paper. That's about as much information as I can.
Okay. All right. Thanks. All right, so Arthur, we've kind of moved from small molecule targeting RNA through delivering the mRNA itself or mRNAs, and now we have your platform targeting proteins. Maybe you can talk a little about that idea of thinking about how you balance risk and think about what part you want to start building your pipeline out with.
Sure. So I think Jake and Jennifer already made some really good points, so I'll try not to repeat some of those, but I echo what they've already recommended about target selection. So we start out by asking ourselves, what advantage do we have with our unique modality? I look at targeted protein modulation and we see technology that allows us to move any protein up or down. I see a slightly confused look on your face, Jeff. Do I sound okay? Okay. Yeah,
that's my intense listening look.
Okay. I was like, I hope I'm speaking English here, but so.
You're loud and clear. You're loud and clear, Arthur.
Okay. Thanks, Sarah. So with targeted protein modulation, we can move any protein level up or down. And so that's a unique advantage. And then we ask ourselves, well, which disease categories and which targets might benefit from that, so that we're not just imitating another inhibitor molecule. We're going to be removing a protein via the ubiquitin proteasome system and via ligase mechanism of action. So we like to ask ourselves, what advantage would our mechanism bring? And that opens up two major categories. The first is targets that are well validated, as Jake and Jennifer say, but may be resistant to current inhibitors. So resistance mutations is high on our list because we can bind to such a mutant protein that escapes today's drugs, and we can degrade it, just simply degrade it with a small molecule. These are oral small molecule agents. An example of that is our BTK program. So BTK, Bruton's tyrosine kinase, is of course a tried and true target for B-cell malignancies, but there are resistance mutations now cropping up in the clinic. And our compound, for which we plan to file our IND in Q1 of this coming year, so in a few months, our compound can degrade that resistance mutation. So resistance mutations in general are very high on our list in target selection. The second major category are the undruggable proteins. So if indeed we can move any protein level up or down, let's tackle some of these undruggable categories. And some of those, are on everyone's list, but they still remain great medical challenges for medicine. And so we're going after those. I should mention there's a third category given our unique approach, which is modulating E3 ligases, which are the business end of protein regulation in the cell. We can inhibit a ligase, a particular ligase. There are over 600 ligases encoded in the human genome. And so our second program that we'll file our second IND on next year is a ligase inhibitor. And this is CBL-B. So we've uncloaked these so I can discuss them. CBL-B is a very unique ligase that normally turns off T-cell activity after the T-cell is activated by degrading proteins within the T-cell, so to turn it off. And so we can inhibit that function and move those signaling proteins back up and turn on the T-cell again. So that's major category is the undruggable or new target. CBL-B is definitely in that category. So that's how we think about, it's really a portfolio approach, as I think you've been hearing, starting out with validated, but then also having really the frontier in front of you of the undruggable targets.
Great. So one thing that I think is kind of a hot topic or trend these days, one of the greatest proponents perhaps, and also a great and eloquent speaker about it, is Dan Chen, about this idea of increased complexity and the smartness of bioengineering to kind of allow multi-component, kind of multi-targeting, basically inherent combinations. It's very clear that where that could stand with Jake. We'll let Jake comment a bit on that, but I'm just curious to what degree either of you are thinking of that inherently within your own portfolio builds, that kind of your own kind of biologic and franchise synergy, as opposed to simply developing an agent and then hanging that on top of an extant standard of care. Obviously, that introduces some risk, but I'm just curious to what degree you've been thinking about that. Jennifer.
Yeah, so we actually give that some thought. It's interesting that there is an emerging set of databases that point to synthetic lethals, which can be challenging for small molecule or other drugs because often are synthetic lethals with novel proteins whose function is either not known or poorly understood. They're endogenous inside the cell. And the principal value for many of these targets emerges precisely because they are synthetic lethal. These are actually very fertile ground for us because you can think of what we're trying to do in some ways as being a small molecule that phenocopies an shRNA. So as we pursue some synthetic lethal targets, we hope to be able to exploit that fairly rapidly by going after underlying RNAs rather than the proteins themselves, which may or may not be druggable.
Great. Good.
Yeah, I can add from our perspective, the targeted protein degradation molecules are bifunctional molecules. So they have a one binding component to a ligase and another to a target. And one can, and in fact, our BTK program, our lead program, illustrates this, actually use the ligase portion to target another protein, another target. In this case, that ligase portion is an imide-like molecule, so a Revlimid or a pomalidomide. So we target and degrade the substrates of the imide. Ilos and Ikaros are the names of those particular protein targets. And then the other end, we degrade BTK. So these are people think about bifunctional, antibodies or bispecific antibodies. These are bispecific small molecules, basically. And so you are engaging more than one target, and you're inducing proximity of targets. So it is another level of complexity for, over and above the traditional small molecule. But that's our take on that.
Yeah. Great, good. And then certainly, plenty of companies working on biologics that are bi or multifunctional. Macrocyclic companies now are working, trying to kind of mimic some of those bifunctional. So then, Jake, you alluded to this before. It's inherent in your system. You talked about, how you might influence the tumor microenvironment, the tumor and or the immune cells. So when thinking about those multi-component type of approaches, again, that by definition increases risk, and you talk about kind of balancing risk. So how are you thinking about that? And are you thinking it from the standpoint of incorporating validated targets, like you could deliver a checkpoint inhibitor? And again, I'm talking theoretically, so I know you don't have to prove anything that may be proprietary at the moment. So
what we're looking at is, how can we, it's not all about what the RNA delivers, right? Because with RNA, what we have found is that all of the therapeutics up until now have essentially used RNA to deliver a protein, right? To encode cytokine target or, any other sort of gene on the messenger RNA and deliver it. We definitely do that part. That is very important to what we're doing. But RNA can be leveraged in a number of different ways. If you look at some of the data coming out of Checkmate Pharma, for instance, with their TLR stimulation, when you look at what people are doing, what Regantech was doing with their RIG-I stimulation, when you look at what oncolytic viruses were able to do simply just by lysing the tumors, when you start to look at all those different mechanisms, what it allows you to do is, when you can actually control and program the messenger RNA to behave in different ways, it allows you to hit multiple different mechanisms in different times. And so what I mean by that is we're using mechanisms that other people have shown in the clinic to be efficacious, such as innate immune stimulation, which traditional mRNA can't really do, because the moment you start simulating those pathways, you turn on interferon and you shut down translation. But what we're doing is we're taking this orchestrated approach, which allows us to push different buttons at different times, that with the data that we've put together is wildly effective. I mean, you're talking, single injections into large, highly immunosuppressed tumors, and we see 80%, complete remission in these mice. I mean, it's preclinical, but it's very promising. And so to me, it's about, how can we leverage the entire system, not just use mRNA as a data carrier exclusively, but if we are able to, use the body's natural responses to these mRNAs, perhaps we can lean into some of the issues that we've seen, such as innate immune stimulation that impacts translation. If we lean into that response and say, actually, I want some of that's a good mechanism for this solid tumor. Now I can build on top of that more mechanisms that are different in scope. And then, as we expand and want to take on more risk, you can take, the cocktail approach, for instance, that others have talked about, where, you encode multiple different types of proteins and deliver them all in one cocktail.
So quick follow-on question. So inherent in the idea of the programmability, and as you're indicating, not just the expression of a protein, but other functions that the RNA can adopt. Are you also building in kind of exogenous ability to control activity?
Yeah, so we are looking at those options. It's, as I said, we keep it simple for the first program and get into the clinic, but anyone who PubMed's my name, or Ron Weiss's name, who is my advisor at MIT and co-founder of this company, will find out that a lot of our technology even lends itself to exogenous control. And so we can get to the point where we can pulse proteins, we can pulse, cytokines maybe, because we have RNAs that respond to small molecules. We can even use a dose controllability, where we use the small molecule dosage to titrate in the amount of protein that's being expressed. And we have found in some early work that could be also very efficacious, especially when we try to, move into other sorts of tumors. Now, I'd love to make the promise that, STX-001, our lead that'll, be entering clinical trials in the next, 18 months or so, I'd love to say that it will be a pan-cancer therapeutic and fix every single solid tumor that's out there, though I don't think anyone can really make that call with their cancer drug. But what my goal is that we can continue to innovate and build new therapeutics for different types of tumors and continue to explore what those drugs are able to do. And we do that both in-house and now also through partnerships with other groups that have, maybe developed some proprietary proteins that have some sort of effect. And now instead of delivering it as a protein therapeutic, we can change it and encode it back on the messenger RNA and directly express it that way. And so there's all sorts of different innovations that I think the platform allows you to do once you take full control of its life cycle and how it behaves in the body.
Yeah, and baby steps can be innovative inherently. So
our lead drug is no slouch. I mean, it's an easy approach for us, but it's completely novel, and it's going to be good.
So I'm going to go to you, Arthur, before I go to you, Jennifer, because some of what Jake was just talking about kind of starts to segue into some of your space of controlling or modulating RNA with small molecules. But before I get to that, actually, I'm curious about this idea of just exogenous manipulation. So, Arthur, I know you've got this new, is it a spin out or a JV or what, the cell therapy angle here?
So Descartes Therapeutics, it's drug-enhanced CAR T therapy, is currently a wholly owned subsidiary of Nurix. And it is, it's the concept of, to put it simply, cells on drugs. So the cells are taken out of the body, and of course, during that period, they're very accessible to targeted protein modulation. And so we're able to adjust protein levels up or down with small molecules. And what we found, especially with the Sybil inhibitor applied ex vivo, is that it doesn't just alter the protein levels transiently, but it results in a reprogramming of the cells towards a more central memory phenotype. And so that phenotype then persists within the cell and in the animal models we've tested so far for months. And then also we can then go back in with an oral agent, talk about exogenous control, and re-stimulate those cells with the oral version of the drug. So we think that applying targeted protein modulation to cell therapy is going to be, a whole new way to modulate cells ex vivo, rather than having to do the genetic reprogramming, which is, reaches a sort of point of diminishing marginal returns. We can only put so much genetic material into a cell. So we think small molecules will, and we have a major TIL effort in that regard too. So for tumor infiltrating lymphocytes can reprogram the same way.
Good. So then to you, Jennifer, so this idea of, using those small molecules to not simply abrogate activity, say as in Nick, but maybe to modulate or even the idea of targeting for you, not the tumor, but other cell types, say immune cells and the TME, are those things that you're thinking about?
So first of all, if you look at the entire life cycle of the RNA, largely speaking, there are exceptions, of course, but largely speaking, translational regulation is in the 5' UTR, untranslated region. The regulation of the lifetime of the RNA is typically located in the 3' UTR. And then, of course, splicing is found in the introns and exons that ultimately culminate in the open reading frames. So each of those offers an opportunity to regulate what the RNA ends up doing. So typically, you would imagine let's just say it's easier to screw things up than it is to make them work better. Let's just face that. But you can imagine that a molecule binding to a regulatory structure in the 5' UTR could lead to either to impeding translation by a variety of mechanisms, or actually to the enhancement of translation. There are examples in the literature where structure, as you strengthen the structure for mutation, that you actually get more translation. Similarly, you can imagine in some cases elevating the lifetime of the RNA by suppressing the degradation, the programmed degradation that takes place 3' UTR. We understand, of course, that just having more RNA doesn't necessarily mean you will get more protein, but it's a good first step. And then finally, in the splicing region, there's a lot of these splicing events are not particularly efficient, and so energetically they kind of go one way or the other. And so they represent real opportunities to nudge the one outcome or another, either to produce truncated proteins or proteins that express the intron occlusion, or ultimately just produce nonsense-mediated decay because of various kinds of stop mechanisms. So yes, there's an opportunity to do real, as they say receptorology here, to look for agonists, antagonists, inverse agonists, where each possible outcome is possible from a given structure lined up with a small molecule. Now, to your question about the cell types, we haven't made a real distinction between going after the actual tumor cells or their environment. In essence, all targets are potentially available to us. So we tend to pick some of the, really heavy-duty, low-hanging fruit. And it's like not only is it low-hanging fruit, but we're the only people in the orchard. So, we can go after transcription factors. I mean, our discovery pipeline is just this rogues gallery of famous targets, because why not?
Yeah, all undisclosed. Target 1, target 2, target.
Yes, I appreciate. I don't want to get out ahead of our team in this regard. What I can say is that in the oncology space, we're talking about MYC fairly freely. In the non-oncology space, we're talking about ApoC3 with some frequency.
Great.
So.
Okay, good. So now let's kind of tie some of this science and biology and synthetic engineering, et cetera, back to some of the kind of commercial business strategic questions. So I'm curious, as you, and this is going to be, I think, some of your thinking very pertinent to some of the NUCOs or more early stage entities that might be listening to us. How have you found it kind of presenting your story and making the pitch around the value proposition of what you can do? It's one thing to say it's novel, and that has historically checked a certain box in various types of investors and pharma, particularly those interested in early stage. Clearly, intractable or undruggable is also kind of a checkbox for many. But I'm just curious, kind of how you've thought about that and what type of reaction you've heard as you've told your story to investors or to partners. Are there particular things? And again, obviously, this is only what you can disclose publicly. Are there particular things that people are latching onto, and are those the things that you expected, or have you discovered interesting things along the way as you've tried to tell the story? And that's, I think, going to be a little different for each of you, and probably it's a much more validated space, at least, Arthur, in the sense that there are a half a dozen or more other entities kind of doing a similar thing, not identical, because there's lots of other ways to do this targeted protein degradation. But so maybe Jake, we'll start with you there, and then Jennifer, and then Arthur. So.
Yeah, that's a fantastic question, actually. So, in the messenger RNA space, there's a double-edged sword, I think, right? Which is, on one side, you can point to the number of multi-billion-dollar IPOs we've seen over the last 2 years, and now into the double-digit billion market caps for multiple mRNA companies, and point to that as a part to say, this technology has legs to grow. But of course, it also raises the question of, there are all of these companies with all of this money, and how do you see yourself fitting into that space? And I think the story that I tell around that and how I believe about it, what I believe, is that messenger RNA, in terms of the first generation of companies that are now mostly all public, those companies all were focused on, can we make messenger RNA work? And how will that happen? How do you manufacture clinical grade messenger RNA? 10 years ago, not even 10 years ago, but at the first ever mRNA health conference that was put on by Moderna, BioNTech, and CureVac, I went as a second-year grad student and was sitting there listening, and everyone was talking about how we're going to make clinical grade mRNA. What enzymes are we going to use? What precautions do we need to take? What are the cap analogs? And all of these different questions that then became answered and opened more therefore, because the market expanded itself, they became open to new entrants as well, right? I mean, Moderna and BioNTech have their own proprietary processes, but there's also a lot of processes and enzymes and reagents and knowledge that is accessible to everyone. And that brings us to what I think is going to be the next generation of messenger RNAs, which is that mRNA itself, its existence, is not the novelty anymore. I mean, if you're a large company with lots of money and a diverse pipeline and a big manufacturing unit, then you have the first mover status to be protected, like those large public companies are. But I think for building new therapies and innovating in the field, it's more about what is the sequence of the messenger RNA you're using, and not just the gene sequence, but the entire soups to nuts design of the messenger RNA itself. Essentially, we're commoditizing the hardware of messenger RNA, and now it's all about the software. We're switching over. What are you using? How are you designing the messenger RNA from every single sequence element? How does that impact the therapeutic efficacy and the sorts of diseases you can go after? And so I think the story from Strand that sticks and is exactly in line with what I've been working on for the past almost 10 years now with my co-founder, first at MIT and now at Strand, is that if we can build messenger RNAs that can, only turn on in target cell types, and we can build messenger RNAs that can be switched off, and we build messenger RNAs that can hit multiple different mechanisms in a concentrated kind of orchestra of mechanistic stimulation, then we're at a new level of messenger RNA. We're talking about the next kind of frontier of how that moves forward. And we're talking about the shift from hardware-based organizations, where we were with messenger RNA and where we've been with a number of gene therapies, to software-based orientation, where the innovation and the IP is driven by how you design them, what that sequence is, and then the RNA itself is essentially a commodity, is a type of commodity. Companies still need to make their own processes, and we take manufacturing very seriously. We take automation in manufacturing very seriously to also decrease the cost of what these drugs will eventually cost us in the clinic and cost us commercially. But to me, it's that mRNA is quickly becoming a commodity, and so how do we then innovate on top of that commodity to serve further value and create better drugs?
Great, perfect. All right, Jennifer.
Yeah, so as we've had some very good fortune talking to partners. For the most part, partners have come kind of pounding on our door, and I think that's because the story very much lines up with things that make a lot of sense to them. And that is that we're going to focus on small molecules, which are a highly validated therapeutic modality. Not that they're perfect, but we're familiar with their imperfections. Second is that it represents a broad new potential range of targets, thousands of possible new targets, and many of them that overlap from a mechanistic perspective ultimately with targets that are already known to be validated and of interest. So it's a matter of just sort of connecting the dots. In terms of how it's been received, it's really been received very well. Again, because I think they're prepared to hear about this and appreciate how it will move in the right direction with them. Also because there's a lot of overlap between our platform, when we're very much a platform company, and their platform, which is that, pharma companies are actually platform companies. So this culminated recently in us doing a fairly substantial multi-target deal with Roche in April. And can't talk about the number of targets or what they are, but a wide range of targets of interest. And so, our approach is to go after this and not only produce the starting points that are important to them, but also to sort of learn with them how best to do this kind of science.
Yeah, and of course, those type of multi-target, undisclosed, large pharma deals are a classic early company value inflecting, because they not only give you a certain validation, but they also help you kind of build up the rest of your pipeline as you're focusing beyond just whatever those undisclosed X number of targets are. So, a lot of history of companies building up that way that are platform companies.
Yes, the value for us is that it gives us the time to enhance our platform, understand what we're doing. It gives us the time and resources to take the specific targets that are our internal targets and advance them as far as we can. So it's highly enabling, I think, not just for Roche, our partner, but for us as well.
Yeah. All right, Arthur.
Yeah, so that was really part of our history, you're right. So, in the early days of targeted protein modulation, there weren't a lot of us. In fact, there was probably only a couple. I think Nurx was one of the first movers or early movers.
Yeah.
And I'd say that companies at that stage go through certain phases, and that phase is sort of the beauty is in the eye of the beholder phase. And in order to find a partner that appreciates what you're doing, they have to have some internal understanding and depth and maybe insight. In that case, it was Celgene for us back then in 2014. And that was, as you pointed out, Jeff, a typical multi-target, big alliance, $150 million up front. And because they understood the image, they understood Cereblon, they understood ligand, they saw it, they lived it, they saw the data, they knew what could be done. Others didn't understand it. I mean, we'd walk into business development meetings and say the ubiquitin proteasome system, and the eyes would glaze over, and that was it. Lost everybody, right? So that was a phase. The second phase, which I guess I'd call the Missouri phase, which is the show me stage phase, and all the big pharma executives who don't have insight in this particular technology. It was, show me the data, show me the data. I mean, endless data presentations and free seminars. And, it finally penetrated because, we had the data. And so once you start to get the data and it's solid, then momentum can build. And now there's really good momentum in the field.
So that Missouri phase, was that as bad? 1 second, Jake. Was that as bad as, the Moses wandering in the desert, kind of lost and alone, or was it not quite that dark?
There was a moment when BMS bought Celgene and we lost our major sponsor, but we still had the $150 million. So it wasn't a bad desert. I mean, we had material to go with us, supplies.
Yeah, can I add something, by the way, here? I was at Celgene at the time, so I was on the other side of the table. And what I can say is that Celgene represented precisely the kind of party that appreciated what Nurx was doing and what they had accomplished. And therefore, it was really a very good marriage, at least, at the time that I was there. And I think that we experienced the same thing, is that if the potential partner already is thinking about the problem, you're 80% of the way there.
Yeah. And having worked for said entity in some interesting way, I think you're right. I think that internal knowledge can be very helpful for one, it gives you some champions potentially, aside from just helping understand. So Jake.
I just want to, what Arthur just said about the show me days and how that, or the show me days of the company. And I think that is exactly our experience in terms of the response that we got when I was, going out and trying to fundraise before the company existed. And I said, programmable messenger RNA will be the future. We'll be able to hit we'll be able to build a therapeutic that does all these different things. And a lot of investors said, why does that, okay, maybe it's like a pipe dream when you're talking the story and you're giving all the scientific reasons, as Arthur says. You're saying oh no, this is the reason this will work, and this combination is able to be done with one molecule or something, and we'll be able to hit all of these different things in such an orchestrated manner. And that was a much harder sell than it is today, where I just say, look at this, this is this is some awesome shit. This is some good data, and I can actually kind of show it off.
And you weren't even bleeped for that.
Yeah, well, I mean, it's a much easier.
I was on mute. I couldn't respond quickly enough to delete to mute to bleep him. So go ahead. Sorry.
Sorry, Sarah. I'll watch my language, but it's exactly like it's this. It's this. I think there's a slight risk-taking aversion, though, in some areas of biotech investing for early stage. I mean, we went to Silicon Valley, and I said, programmable nucleotides are the future, and they were like, yes. But in some areas of biotech, I was like, well, programmable nucleotides, programmable messenger RNA will be the future, and people were like, well, show me, and I was like, well, give me the money, and they were like, no. And so it's like cart before the horse, and it's a storytelling exercise as well. But it's definitely kind of I just when you said that, Arthur, it just hit me right there in the chest.
Right. So I have many more questions, but I know we're coming up at 10 of the hour, Sarah. So what do you want me to do?
How far off are we? Oh, we are at 10 of the hour.
We are at 10 of the hour. I watched the clock.
Okay, so Jeff, let me say this to you. Pick your best next question, then we'll move to wrap up, and we'll go into Q&A from the audience. Okay?
Oh, all right, that was cruel. All right, I suspect we'll get into some of the questions, but let's go with this one, because this actually is one of the questions that came in before, and we haven't really, you've kind of touched on it a little bit, Jake, but we know that this has been the age for a while of immunotherapy, and although we're seeing a little bit of retrenchment and a little bit more re-emphasis back to, kind of the idea of precision oncology, which is kind of what was hot before the age of IO, it does kind of raise the question of how are each of you thinking of kind of biomarkers and companion diagnostics and patient stratification. Now, for Arthur, it's kind of more inherent to some degree in what you're doing, but not necessarily clearly as so for you, Jake, and for Jennifer, obviously their expression levels of MYC or relevant importance in different tumor types, but maybe you can each kind of briefly talk about how you're thinking about that, or to what degree that's kind of entering your thinking as you're envisioning kind of that stage within clinical development. So let's start with you, since it's inherent, and then we'll quickly go to Jennifer.
Yeah, I'll be brief, but illustrating with the BTK program again, it's really great because you take this oral molecule, and then basically we take a series of blood draws and we watch BTK protein disappear from the bloodstream by FACS analysis or Western blot. It's very satisfying science. It's very, very much a proximal biomarker, the most proximal. And then, of course, we layer on the downstream biomarkers. But watching protein levels, either disappear and go away or go up, very satisfying results in biomarkers.
But a quick big quote-on question to that. So presumably, whether it's BTK or things like EGFR or RAS, you could because of how targeted protein degradation works, you could be all-encompassing in terms of hitting the protein and the multiple different mutations that a given target protein has, right? You just pull down the whole protein.
Yeah, the whole thing goes away, and you don't need high affinity binding either, which is often the issue with resistance mutations, is they abrogate binding of some type.
Yeah.
So the whole protein goes away, and literally within 2 hours, 4 hours, 8 blood draw, that's going to be our biomarker in phase 1A, and you just watch the curves go down, and then it'll help us pick a dose and help us understand the PK/PD really in a very proximal way. And then, of course, you have your downstream biomarkers that are more related to immune system activity that we monitor, but everybody can monitor those. But I really love the targeted protein degradation side of that biomarker story.
Great. Jennifer?
Yeah, there's a lot of analogy, actually, between what Art and his group are doing and what we're doing, in that, I think of targeted protein degradation as fire suppression, and we're doing fire prevention. We want to prevent the protein from ever having existed. And so similarly, we would, this will be, of course, strongly indication dependent, but in indications, for example, that have liquid tumors, you can imagine administering the drug and looking at least one type of biomarker is the diminution in the expression of the protein. I suppose that we could also do a parallel assessment of the RNA level for that protein as well. So I think that's actually relatively straightforward. There are, other markers. It would just be completely target dependent. And I would say also patient selection. We would be interested in making sure that the sequence of RNA that we were targeting, and therefore the structure that they made, was actually likely present in that patient.
Yeah. Okay. And Jake.
Yeah, I mean, for our patient selection, we're taking a much broader approach, as our goal is a kind of off-the-shelf therapeutic that can be applied across a number of different solid tumor types. So we are stratifying the types of tumors that we would eventually like to go after that we think would be more amenable to these sorts of therapeutics. But for the most part, we're monitoring things like a number of the immune markers that Arthur was alluding to, as well as, our protein and certain immune responses that we know, such as like interferon stimulation and such. We also need to, I've been clear that we are expressing a cytokine from our drugs. So I think with cytokine drugs, it's important to monitor the local as well as the systemic expression of both of them, and make sure neither is getting too high, as that's one of the larger risks that you can run into when you're handling these sorts of things.
Yeah, great. Okay. All right. Thank you. So, Sarah.
Well, thank you all. Thank you all very much for spending your time with us this morning to share your insights. Jeff, thank you for leading a great discussion. We are going to move on to the Q&A section. So again, for those of you who do have to leave, a complete audio tape will be available on our website in probably 3 hours. And any available documentation will also be posted there, as well as the schedule of our upcoming virtual salons and fireside chats. So do join us for those. I would be remiss if I did not thank our sponsors and our supporters. They are really very important to keeping this program going. So now, if you don't mind, we're going to move into the Q&A section, provided I can open this up so I can see them all. Oh, okay, we have quite a few. So the very first one is, how can we de-risk emerging cell and gene therapies for cancer if funding for early stage programs, for example, in GBM or PANC, is hard for companies to come by, too risky to get funding and to make it into the clinic? That's one question.
That's a great question. I can take a biased answer. I think that cell therapy in particular is very difficult right now in terms of scalability, right? And it's a massively expensive cost to move any therapy forward. So before I like to think about how do we, build more cell therapies, I try to think about how do we make the current cell therapies more effective. Things like allogeneic approaches, I think, are going to be huge. Off-the-shelf sort of therapeutics that can be expanded to new sorts of indication areas, or some of the approaches that are now emerging to deliver CARs to T cells and NK cells in vivo. Right? Can we make these can we turn cell therapies into just drugs that we're delivering? And so, as with everything, in a perfect world, we would fund every avenue of research for these sorts of areas. But I think we have to, in order for cell and gene therapy to go after those tumor types that are mentioned in that question, GBM, pancreatic cancer, where investors are very hesitant to move into, we have to start thinking about how can we make those therapeutics, those gene and cell therapies, for much lower cost, and then we can hopefully apply them across different sorts of tumor types. I also think that the recent economic plan from Joe Biden provides a lot of federal support for those sorts of higher risk research. And I think that the government, or I think the country and really the world would benefit greatly if the government stepped in and provided more support for high risk research, not just at the discovery phase, but really, funding through the valley of death into a commercializable product. And for something like pancreatic and GBM, that's even much further than what we currently have. And right now we have still a big valley of death between academic research and startups. So we need to find ways to bridge that. And I think that, the federal government and other sorts of large bodies like that can support that research in the name of the public good will be a huge piece of that moving forward.
Oh, great. Thank you so much, Jake. Does anybody else want to comment on that? Okay, if not, we'll move on to the next question. What are the undruggable protein targets that are the most important? This comes from Arizona.
I think that's queuing up Jennifer and Arthur in particular.
That's a big question.
Well, it's a little open-ended, but I think that there's a lot of consensus around the kinds of targets that are difficult to drug. And let's just take as a class transcription factors. Transcription factors have been around for a while. They're very well understood for the most part and what the implications are. You can watch them be modulated by drugs that don't address them directly, but rather indirectly. And so they clearly play very important roles in the life cycle of the cell. And for many people, there's been a lot of effort to go after them more sort of confrontationally, and we are now among them. So they are not. So far, drugging transcription factors has proved to be a very, very difficult and unrewarding pathway. I have concluded by looking at RNA structures that drugging RNA structures has got to be easier than drugging those darn transcription factors. And so that's really our approach to this. But, I'm not sure that I would go through a sort of name the rogues in the rogues gallery of targets.
I would add to that, if I could, just that echoing a certain class would be the mutant oncoproteins that we've known about for decades and haven't been drugged yet. I mean, they're just really have to be addressed efficiently. I then also turn to the protein aggregation diseases, especially neurologic, that have remained beyond medicine's abilities, apparently. So I think that's another category. And then I think the third is a new one that we're now painfully aware of, which is viral proteins, I think can be approached with non-traditional methods, and we can create new classes of antivirals. I'd put it for me, those three buckets are the ones.
Okay, thank you. Thank you, Arthur. Jake, do you want to add to that?
I would not speak nearly as educated on undruggable protein targets as the two of them, so I can leave it at Arthur's final word.
Okay, and okay, Jeff, any comment before I move on to the next question?
No, I think Jennifer and Arthur laid it out well without going through a long list of the known targets. The known undruggable.
Okay, so here's another question that I think gets to a little bit of the access discussion we had yesterday. The question is, once manufactured, would these therapeutics be easily leveraged in any setting by type of oncologist or hematologist, i.e. by a community oncologist in an outpatient clinic to an in-hospital setting? Wow, okay.
At least in our case, we expect them to be orally available small molecules that you swallow. So I should think that from a logistics standpoint, from a clinical practice standpoint, we would expect the answer to be yes. It's not a procedure, it's a prescription.
Yeah, but I guess we might want to have the caveat to the question that, it's not just the modality, it's also the target and therefore the AE profile that also dictates, how readily these move into the hands of the community oncologist from the major centers. So that doesn't apply just to you, Jennifer, that applies to everybody.
Yeah, fair enough.
Okay. Okay, thank you, Jennifer. And Jake,
Yeah. I mean, this is something that I've been, very passionate about, and why I really am in, messenger RNA and programming messenger RNA as a field, because I feel like a lot of the gene therapies and the cell therapies, for that matter, of what we've developed so far to attack cancers is just incredibly hard to scale to a community hospital setting. Even like the cancer vaccine technologies, the bespoke cancer vaccine technologies, you have to be able to you have to have the infrastructure to sequence and then remanufacture bespoke mRNAs and get them out to people. And so, in my dream future, for all sorts of cell and gene therapies is all built on, just modalities that can simply deliver genes in a simple way. Systemic delivery, I think, is where we're going to have to go to get to, full penetration into community hospitals. It's not as easy as an orally available small molecule, as Jennifer had just said, but the idea of an IV drip of of an engineered gene therapy that can then only specifically be activated inside of a tumor, I think, is how we get to wider groups of patients. Same thing with cell therapies, either allogeneic or in vivo generation and delivery of a CAR to a targeted immune cell population. That's how we get to more people and into a community setting, in my opinion.
Okay.
I'd like to add, could I add something to that?
Sure, go ahead, Arthur.
I mean, every major innovation in medicine has faced this hurdle, and they have something in common. If these innovations work, if they really provide value to the patient, the barriers will slowly come down and the technical hurdles are overcome. I can remember when bone marrow transplant was done, and only very few centers would embark upon it. I mean, you could go right down the list. Farther back, open heart surgery was unheard of and, outside of the major medical centers. And then I remember when CAT scans came, I'm dating myself here, but when CAT scans, people were all like, how are you going to get a CAT scan, rationing CAT scans? Now they're everywhere. So if they work, we find a way, I think, and it does take time, though.
And I would just add to that is that it's not just, so what we're really talking about is value, which is what you were alluding to, right? Yeah. So whether there are ways to properly address the democratic access for everyone via different pricing models, that's kind of one angle. The other side, which everyone's kind of alluded to, is technology advances at quite a pace. So while it's one thing to have this discussion about how readily accessible to a community center and to the patients going there is an autologous CAR T, it's a very different question, although, it's not to say that it's, a low price, but it's a very different question if it's an off-the-shelf allogeneic type of cell therapy. So again, technology and those innovations can walk hand in hand with the more, let's call them political and institutional types of innovations that can kind of help seed the way.
Okay. Thank you all for that. Our next question is, actually it's directed at you, Arthur. Does going public influence your partnering or R&D strategy for all the public to hear, based on what public investors appreciate versus early stage private investors?
Well, definitely there's an effect. I mean, in the early stages, you have really only two sources of capital. You have partners and you have investors, and that's what you have. So these are your customers. And so we're in a phase where our customers are our investors, primarily at this point, I would say. And depending on the amount of capital that one can raise, what it does is it allows the company and the investors to own the lead program rather than partner them off or do dilutive types of partnerships. So it tilts it towards an internal pipeline and making one's own choices to further the development of a wholly owned drug, ultimately. So that does influence your thinking. Some of the other partnership programs may be longer term, higher risk, and they're better to have with a partner. But with a strong investor base, the company can make decisions on its own and own those assets for the investors. It's a great thing.
Great, thank you. Does anybody else want to comment on that, even though it was directed at Arthur? Okay.
Ask them in 3 to 5 years from now.
Okay.
Maybe even sooner, actually, at the way things are going with the IPO. Who knows? Could be a year from now.
Yeah, Jake, I can't believe you haven't done your A round yet. Am I correct in thinking that I heard correctly?
Anything.
Oh, okay. Okay, that's another
there's
story.
There's lots of opportunities on the table. I think we talked about earlier, and actually what Arthur just talked about is all the different sources of capital that are widely available because of interests, and you have to balance the financial interests of owning something fully for the company through investment dollars, but giving away a piece of your company versus, giving away the assets in exchange for quote unquote non-dilutive financing, and a number of other very interesting new hybrid models that are starting to emerge, and the sorts of collaborative development that you can do there. But we're always while I come on these things and talk a lot about my views on the development of drugs broadly, I think we're always a very quiet company when it comes to other sorts of those things, at least for now.
Yeah, although I would say implicitly your desire for control, and I think it's clear.
Yeah.
Well, we look forward to you jumping out of the quote unquote box, Jake, when it's
I'll jump out of a cake, yeah.
Yeah, so, okay, with that said, I know we do have a number of other questions that are in here. We're going to see if we can try to answer them offline, but we have come to the end of our time, with our great panelists. I do want to thank you, Jeff, for leading such a great discussion. Jake, for being a part of it. We adore you. Arthur, we adore you too. Jennifer, it is great to meet you. You're part of the pack now. So
yes, thank you. I want to echo what Sarah said. Thank you, everyone. It's great to be, this is dial number 4 with Jake, and hopefully, many more coming up with Arthur and Jennifer. So.
And Sarah too. And Sarah, we're going to do more.
Well,
So I'd like to thank everybody who has stayed on with us. As you sign off, I know there will be a Q&A. Please do take some time to give us some feedback. We know that we had audio problems, so hopefully in the next salon, those will be fixed. We won't have them in the future. And once again, I'd like to thank our sponsors for supporting us. And our next salon is actually a fireside chat with Mike Gato, who is the global head of healthcare at JP Morgan. So he'll be with us on September 23rd. So please sign in for that. With that said, thank you, everybody. Have a great afternoon. Thank you all.
Thanks, everyone. Stay safe.
Thank you.
Bye now.
Bye-bye. Bye.